Hi, good morning, everybody. I know a lot of you guys, but for those who haven't met me before, I'm one of the clinical fellows in Gyne-Onc. And I am today presenting on a rare ovarian subtype on ovarian carcinosarcomas. And really the two goals of my talk are to emphasize that P53 status on IHC is actually predictive of outcomes, and the other thing is for all our ovarian carcinosarcomas to have P53 IHC, and whoever is a P53 mutated be referred for genetic testing. In terms of ovarian carcinosarcomas, I think most of us really look at ovarian cancer and look at like 5 distinct histotypes. And then there's very small percentage of cases that don't really fit into that criteria. And ovarian carcinosarcoma is one of those subtypes. And for the last, 2 decades or so, we know that the carcinosarcoma is sort of a misnomer because they're not really sarcomas. They're actually metaplastic carcinomas that actually go through some transdifferentiation. And a lot of studies, and most highlighted here is one by Dr. Coble and one by Dr. Hollis, really looking at the pattern of P53 mutation and the Wilms tumor WT1 expression, which is really a hallmark of serous histology. Looking at those two, about 80% of these ovarian carcinosarcomas have that particular pattern. And when you look at some of these molecular similarities between the ovarian carcinosarcomas and high-grade serous ovarian cancers, there's lots of them, lots of similarities. Yet the clinical course as well as outcomes for carcinosarcomas is not good, is not comparable. So we looked at 2 cohorts in this study. So our goal was to better understand the prognostic and therapeutic significance of TP53 status and BRCA1 and 2 status, and really evaluate the outcomes and highlight some of the findings as far as treatment in context of PARP inhibitor in the last few years. So this first cohort, I know the sequencing data on one of the slides is small, but really to highlight these are about 30 cases that have IHC and targeted sequencing from a single institution, which made our molecular cohort. And to highlight 2 points is there are, out of those 30 cases, 26 of them actually showed TP53 mutation. And about 4 cases had a wild type TP53 status. And really the mutational profile was very similar to endometrioid or clear cell ovarian cancers with like KRAS, ARID1A, beta-catenin mutations. And in general, looking at the sequencing and the IHC was fairly, there was good concordance, 100% concordance between the sequencing data and the P53 IHC in this particular cohort. And then we looked at a second cohort, which was cohort B, really to highlight some of the clinical data and sort of clinical highlights. And these are cases from 3 different institutions across Canada over the last 6 years or so. So we identified 67 cases that were expert reviewed by a GYN pathologist. And out of those, 57 actually had P53 IHC data. And then they also, again, split into 2 groups. 10 cases were P53 wild type on the carcinoma component, and then P53 mutated over 47 cases. And out of those 47 cases, 7 cases had BRCA1 and 2 mutation, which is roughly 17 to 18%. Similar when it comes down to percentage of BRCA1 and 2 mutation in high-grade serous cancers. And one of the cases with endometrioid histotype in the wild-type group had mismatch repair deficiency and a known Lynch syndrome. And then we had one clear cell in that cohort. And then none of the patients had BRCA mutations in the p53 wild-type cohort. Again, small numbers. These are the baseline characteristics of the cohort B. Again, you have to understand that this is a rare cancer. So don't have enough numbers for multivariate analysis, but on univariate analysis, looking at differences in these groups, looking at BRCA wild type, BRCA mutated, p53 mutated versus p53 wild type. The only difference we saw was the age. In the p53 wild type patients were younger compared to the p53 mutated patients. And that was statistically significant. And then the actual, the p53 status and BRCA1 status, looking at prognostic significance, the median overall survival in the p53 mutated, we call high-grade serous-like cancers, was significantly longer compared to the wild-type group, 44 versus 9 months. And this was statistically significant. You can see that on the Kaplan-Meier curve, looking at the survival using log rank test. And then on the other side, looking at the p53 mutated cancer split into the BRCA wild type versus BRCA mutated, you can see the survival difference again emerging. All patients were alive at 3 years in the BRCA 1 and 2 mutated group versus only 50% of the BRCA 1 and 2 wild type. And then out of those 6 of these patients received PARP inhibitor. To conclude, we think that most of the ovarian carcinosarcoma cases have very much the morphologic and the molecular profile that's resembling high-grade serous. However, there's a small percentage of these cases that likely originate through a non-serous oncologic pathway. And this molecular distinction might actually have some prognostic and really treatment predictive implications, especially in the PARP inhibitor era. And then again, these findings underscore the importance of routine p53 IHC in all cases of ovarian carcinosarcomas and BRCA1 and 2 testing for those who are p53 mutated. And then lastly, again, I want to acknowledge Dr. Carey, who's my research mentor, and Dr. Lee from Edmonton, and the entire Carey lab, as well as Dr. Pors, Dr. Coble, Dr. Covens, and Dr. Hopkins. And then many patients who actually consented for their tumors to be tested, and then funding. Thank you.
So it's still morning. Good morning, everyone. And thanks for GCI committee for giving me this opportunity to give my talk on my PhD project with the title of Human Endometrial Organoid Models Implicate G6PD Dependent Metabolism as a Potential Targetable Pathway in ARID1A mutant gynae cancers, including CCOC. Just a brief introduction. CCOC is a rare epithelial subtype of ovarian cancer, which accounts for about 12% of all ovarian cancer cases in North America. It is inherently resistant for chemotherapy, but unfortunately, main treatment option for the patient is still. It has clear cytoplasm due to accumulation of glycogen, which indicates altered metabolism in this subtype of ovarian cancer. It is strongly associated with endometriosis, mentioning that the cell of origin for this subtype of ovarian cancer is endometrial epithelial cells. And ARID1A, a subunit of switch-SNF chromatin remodeling complex, has been mutated in 50% of the cases, often together with PIK3CA activating mutations. Why we raised the question to study clear cell ovarian cancer and ARID1A mutation in this subtype of ovarian cancer? As I mentioned, ARID1A has been mutated in 50% of the cases. And it's been found by our lab and other groups that it's an early event in cancer development because the non-cancerous lesions close to the cancer sites also showed the mutation with ARID1A and PIK3CA. But interestingly, there is no clinical relevance for the patient with the tumor grade with the mutant when you compare the mutant and non-mutant cases. So we think that there is something happening in the cancer development with ARID1A mutation. And if we study these early initiation events, we may found treatment options for the patient with this subtype of ovarian cancer. And in this project, we aim to use organoid culture to study early initiation events, and we hypothesize that ARID1A mutation results in temporal epigenomic and transcriptomic alterations, and studying these alterations which may provide treatment options for the patient. As I mentioned, the cell of origin for this subtype of ovarian cancer is endometrial epithelial cells. So we get endometrial tissue from non-cancerous patients from VGH, and we dissociate these tissues with a specific protocol developed in our lab and put it into 2D culture to increase the proliferation of progenitor cells. Then with the lentiviral plasmids, which are cloned ARID1A guide RNAs and PIK3CA activating mutations, I do the mutations into the culture, and we put it into 3D organoid culture to study the mutation effect over the time. And as you can see, we've been able to create mutated organoids out of these genes, and seeing phenotypic changes in these organoids, we decided to do single-cell RNA sequencing and ITAC sequencing as being part of Switch-SNP chromatin remodeling complex. It alters chromatin structure. To study the transcriptomic profiling changes on the genes affected by these mutations over the time. And we came up with a list of genes, and one of the interesting genes was S100A4 to be upregulated in the mutant organoids compared to the non-mutant, which was validated in ITAC sequencing data as well. And S100A4 is a non-metastatic cancer gene. It doesn't have enzymatic activity. It interacts with the other protein to promote metastasis. There are clinical trials with these genes ongoing right now for colorectal cancer. So the second step for me was to validate the protein level of this gene in the models that we had, like tumors. We were able to validate these data provided by Dr. Cochrane from our lab. And in this endometrial tumor, we were able to show that ARID1A mutant cases had higher expression of S100A4 protein, and you can see a very nice picture of a heterozygous patient with ARID1A mutation, which was reversely stained for S100A4. So it was very interesting for us to see this. And the second step for me was to see whether it was the same case, the scenario in my organoids, which we did the sequencing experiments, and we were able to see the higher expression of S100A4 in IHC staining in mutant organoids compared to the control ones. Also from the cell lines from clear cell ovarian cancer and endometrial cancer cells, because both cancer types have higher mutation rates for ARID1A, we were able to show the inverse correlation between ARID1A expression and S100A4, and it was the same from patient-derived tumor tissues in TMA analysis for endometrial cancer and clear cell ovarian cancer patients. So we wanted to check what is the functional role of S100A4. I made a CRISPR knockout of the cell lines, and we noticed that it decreases the viability of the cells over the time when you knock out S100A4 in ARID1A mutant cancer cell with higher expression of S100A4. And when I overexpress the gene on primary samples, I noticed that it increased the proliferation growth pattern of the cells when we did in-queue site analysis. And we also performed scratch assay, which is an assay to monitor the migration of the cancer cells. And when I did the scratch assay on overexpressed primary cells and also mutant CRISPR cell lines, I noticed that it increases the migration of the cancer cells over the time. So as I mentioned, S100A4 is a S100A4 does the role with interacting with the other proteins. So we wanted to check the interaction partner of the protein, doing immunoprecipitation followed by mass spectrophotometry in 3 ovarian cancer cells and 1 endometrial cancer cells. We noticed that G6PD was one of the candidate genes. G6PD is a rate-limiting enzyme in pentose phosphate pathway. And it provides NADPH to the cancer cells under oxidative damage stress. Our initial analysis with a collaboration with Dr. Rahman lab, we noticed that there is a decrease in the metabolites called glutamine and serine, which are metabolites for cancer cells proliferation. When we inhibit the S100A4 knockout cells with G6PD inhibitor, and my early experiment analysis of G6PD inhibition in S100A4 knockout cells compared to the CRISPR control cells, I noticed that it inhibits the growth of cancer cells over the time. So this is my conclusion. We concluded that S100A4 has been upregulated in our ARID1A mutant organoids and tumor and cell lines. It increases the proliferation of the cancer cells and migration of the cancer cells. And we found an interaction partner in 4 different cell lines doing IPMS experiment called G6PD, and we think that understanding more about the biological function of S100A4 and G6PD may lay the groundwork for the finding treatment options for the patients. So at the end, I would like to thank you for all the members in this picture, especially my supervisor, Dr. Hansman, and Dr. Don Cochrane, and yeah, everyone that I'm missing in this picture also, and our collaborator there.
Hi, everyone. My name is Farhia Kabeer. I'm postdoctoral fellow, like she mentioned. I'm working with Dr. David Huntsman and Dr. Eva Drew. I am interested in studying high-grade serous ovarian cancer and tracking the response to therapies in plasma as well as in tumor at single-cell level. So actually, the overarching goal of my project is to develop a framework for understanding the treatment response and the development of tumor resistance towards the therapy, so we can predict some biomarkers in response to these therapies. Yeah, this is my very old slide, but I really like to show it because it's the simplest way to show like how the clones, they evolve over time. So when I say the word clone, I mean the group of cells that are related to each other by descent through a unitary origin. So this is like, the picture is showing like how the tumor evolves over time, like different colors, different clones, they have dynamical changes. But what happens when we intervene in the clinic with different drugs, the tumor architecture, tumor composition, it changes over time. First, the tumor, it shrinks down, but the tumor, it comes back with full resistance. And I'm trying to show that with different colors, that different clones, they expand, they evolve over time under the drug pressure. So this is, we need to understand what type of clones they are and how can we identify these cells. It could be their mutation profile, it could be their expression, it could be their epigenome, that we can track them and identify them. So being interested in high-grade serous ovarian cancers, we know that high-grade serous ovarian cancer, they have highly aberrant DNA copy numbers. If you look at the plot on top, this is the copy number profile from low-grade serous ovarian cancer, and you can see the quiet genome. There is not a lot of changes in the copy number as compared to the high-grade serous. In the lower button, it's highly heterogeneous with highly aberrant DNA copy number. So can we take copy number as a marker to track the response down along with the changes in mutations? So taking all these things into account, we hypothesized that clonal dynamics of high-grade serous, if we try in an experimental setting of patient-derived xenografts under pressure of different chemotherapies of our choice, and if we track the same in patients' plasma, can we get some potential biomarkers for treatment response or predict any tumor resistance later on towards those specific therapies? And we know still conventional chemotherapy is the first line of choice in high-grade serous, for example, platinum compounds. And we also have targeted PARP inhibitors for the tumors that have homologous recombination deficiency. So in experimental settings, I am focusing more on these two drugs. So how I'm approaching my hypothesis is I'm taking different molecular subtype patients group. I cannot see my cursor, but different type of molecular subtype. It could be BRCA mutant, high grade. It could be HRD, not non-HRD, non-BRCA, or non-BRCA or HRD, different combinations. And I'm taking these tumors where patients are never exposed to any kind of neoadjuvant chemotherapy. So the tumors, they haven't exposed to any chemotherapies. So I am dissociating patients' primary tumor into single cells and looking for copy number profile and this is at the schematics. And at the same time, the same tumor I am transplanting in my immunodeficient mice, and I'm treating those mice with either cisplatin or olaparib or both of them combined. And then after treatment, I'm taking those tumors and doing the same thing that I did with patients' primary tumor and looking for single cell whole genome profiles and their copy number changes. In this way, I can look for the changes and match from the baseline patients along with my patients' derived xenografts. At the same time, from the same patient, I'm collecting blood at the time of diagnosis, it's ctDNA, means circulating tumor DNA. It's the same cell-free DNA that Aline was talking about in endometrial cancers. So I'm doing that in high-grade serous ovarian cancers and taking these samples at different time points, after chemotherapy, post-surgery, and then looking for the tumor burden. And we are also hoping to take these as a tool to non-invasively discover HRD signatures in those patients. We are still in these things. So this is the experimental setting. On this screen, I'm showing you the first results where this is the heat map of all the single cells that are coming from untreated, treated with cisplatin, olaparib of different combinations. So we pulled together all the single cells. So here, each row is one single cell and each column is a chromosome. So you can see they are grouped together by bioinformatically, and you can see clone P and Q, they are grouped together, and clone M, these cells grouped together. And if you look at the marks on the right-hand side, I can tell you, if you cannot see that, these clone P and Q, and clone M, they both are related to specific treatment strategy that I applied on my PDX models. And in clone M, you can see the changes in copy number, actually the whole genome is doubled. And it is the real genome doubling that comes where in the tumor where I treated cisplatin followed by olaparib. So, and at the bottom, there are names of the different genes that are present at those positions in the chromosome. We are digging more into these to find if we can discover more biomarkers. Then I looked at the ctDNA, and you can see the ctDNA profile on top, and you can see the changes in the copy number at different chromosomes. But I was curious, is it the same in the patient's tumor? And you can see this is the patient's primary tumor, and you can see the same chromosomes representing the same changes like we saw in the circulating tumor. And I was definitely curious if I can see the same things in my PD axis because I'm treating them. And interestingly, I found the same thing in passage 1 and passage 2. So I was quite confident that the structures I'm seeing in ctDNA and the mutational, the changes in the copy number is consistent in primary patient as well as the PD axis. And when we look for the clonal prevalence, they were quite similar. Luckily, this patient is Dr. Drew's patient, and she was willing to give us the follow-up sample after niraparib. So she was a BRCA2 mutant patient, and Dr. Drew put her on niraparib maintenance therapy. And the bottom plot is post-treatment. And you can see the changes, tumor burden is decreased, but we can still find the changes at the same chromosomal position. So we recently got this data, and we have around 8 different patients where we have the primaries and the ctDNA. We are following these up. With that, I would just conclude that, yes, we have found changes in evolving copy number in different chromosomes. We can detect large gains and amplifications in ctDNA. And for future, we are trying to get more PD access, and we can see like how much we can correlate those findings with patients' original dynamics. And also, I'm very interested in looking at the RNA, if it's matching back to any expressional changes at the RNA level. With that, I would really like to thank everyone in Dr. David Hensman's lab and in Dr. Yvette Stew's lab. And I will like to say my special thanks to Dr. Andrea Roth and Brendan Lynn for doing all the bioinformatic analysis and everyone that helps us, and especially the patients. Any question?
Hi, everyone. I'm going to talk about Boltzmann semantic score, a semantic bridge between large language model and large vision models. I know this is a bit different and the title is different, but I'm trying my best to explain it. So a little bit background. We know that recently people are using large vision models for AI-driven biomarker discovery, and these models are basically the model that process image data. And the workflow is like this, that we pass a slide image to a large vision model, which is an AI model trained on histopathology data. And then we get the model's output with some highlights from the model. And we then pass these outputs to pathologists and say, hey, this model is identifying some biomarker. Does this biomarker make sense to you or not? So sometimes the pathologists go over the slide. Sometimes they need to go over the past reports and see if there is any significant pattern there or not. But in majority of the cases, there are patterns that the AI is picking, but it's not known to the pathologist. Pathologist cannot really read them. So it's like a black box to us. So there are other issues with this workflow. One is that it's labor intensive, and it's a time-consuming task for the pathologist because sometimes the pathologist needs to go over 100 or more than cases to confirm something. Also, this is subject to bias because it depends on the pathologist's training and background. And the overall workflow is a bit subjective because there is no objective tool to identify these patterns. So with these problems in mind, we came up with a workflow based on the new breakthrough in AI, which is large language models. So large language models are like ChatGPT or Llama that we have in different meta platforms. These models are really good in processing text, especially long and complex text. And we have also, for each patient, we have the past reports, which is basically a detailed report of a case written by a pathologist. And these reports are a set of evidence that supports the diagnosis for the case. So we have the image and we have the reports. And we have now the tools to combine them. So the idea is that we have the slide image, we pass it to the large vision models, and we find the biomarker, which is uninterpretable for us. And then we have the past reports. We pass the past report to a large language models to extract the key information. And at the next step, we try to correlate the key information that the large language model extracted and the biomarkers that the large vision models identified. This way, we have correlated key information plus the visual biomarkers, and now we can pass them to the pathologist for validation. So this is like an easier way for the pathologist because we know what information is correlating with the AI's visual biomarkers. So now they are interpretable. So to do this, the first question is that can these large language models understand and represent past reports properly or not? So to answer this, the short answer is yes, they do. But the longer answer is that we have done extensive experiments on the entire TCGA cancer set with 32 cancer types, over 9,000 patients. And we saw that the model is doing really good in a retrieval task, especially on gynae cancers. And the other task, we wanted to go over the retrieval task. We went to a clinically relevant task, which is patient survival prediction. So in this task, we use the large language models to predict the patient outcome. And again, without any training, I emphasize here that these large language models haven't seen any pathology reports before. They just encoded them. They could predict the patient's outcome. So this basically proved that we can use them to encode the past reports. Then by confirming this, we came up with a method from quantum mechanics, and the name Boltzmann comes from quantum mechanics, to basically match the large vision models and large language models. And then we, based on that score, we first, for a task, we use the large language model as a pathologist to evaluate different large vision models to see which large vision model has the most similarity to humans in evaluating slide images. But beyond this, we wanted to see if we change the large language models from this one to the other one, is there any agreement between large language models or not? And we measured this by kappa score, which basically means that if it's more than 60%, it's a substantial agreement. And voila, the agreement between different large language models is substantial, and sometimes it's almost perfect. And this is this basically heat map is showing on average on the entire TCGA data set. For different cancer set, the agreements can get up to perfect for different cancers. And then this gets interesting when we wanted to find if there is any correlation between the past reports key information and the vision biomarkers or not. So we conduct a correlation test based on two-sided Pearson correlation test, where we try to match the visual biomarkers to text key information. And we saw that for some cancers, especially endometrial cancer, there is a significant correlation between the key information and the AI's biomarker. So basically, what does this correlation mean? When it's significant and it's positive, it means that AI is picking something too similar to humans, but there is cases that it's essentially zero or negative. What does it mean? It means that AI and humans are in different direction. So meaning that there is something in the slides that AI is picking, but humans are not picking, or the other way. So this is basically an idea to automate this process. Now we can identify the cancers that we need to correlate them with human or not. We need to use the AI to find the biomarkers. So the main takeaway of this study was basically showing that LLM can encode the past reports, and also they can measure the correlation, and we can identify cancers that need more a study on them. And the future work is basically we want to identify the exact keywords for each biomarker to pass them to pathologists for further validation, and also apply the pipeline to off-care tumor bank. And at the end, I want to thank for your attention and for the GCI for this opportunity to present my work.
Hi, so my name is Rebecca, and today I'm just going to give a brief talk on our lab's current efforts in identifying new therapies for dedifferentiated endometrial carcinoma, or DDEC. So DDEC is a rare endometrial malignancy that mostly affects postmenopausal individuals. It is histologically characterized with a differentiated low-grade endometrial adenocarcinoma component and an undifferentiated component. And this is an aggressive disease where stage 3 and 4 disease-free survival is around 10 to 40%. Despite this, most patients with DDEC are currently treated with chemotherapy, surgery, and radiotherapy, highlighting a need to develop new treatments that specifically target this disease. So to develop new treatments, our lab decided to focus on a characteristic that is found in a third of all DDECs, which is ARID1A and ARID1B co-inactivation, typically found in the undifferentiated component. ARID1A and ARID1B are subunits of the switch sucrose non-fermentable complex, or the Swiss-SNF complex, that typically binds to DNA to regulate gene expression and repair DNA. When ARID1A and ARID1B is lost, as in the case in a third of all DDECs, this complex is non-functional, resulting in altered gene expression. Based on this information, our lab hypothesized that there can be unique genetic dependencies in ARID1A and ARID1B dual-deficient cancers that allow us to develop new treatments for this disease. So to find new therapeutic targets, we used two methods. The first was to analyze publicly available genome-wide CRISPR knockout data from the cancer dependency map, or DEP map, where for each gene in each cell line, there is a gene effect score, a measure of how essential the gene is for that cell line. A negative gene score means that the gene is essential, while a positive score means that gene knockout increases the cell's growth. For our study, we're interested in comparing the gene effect scores between cell lines with ARID1A and ARID1B mutations and cell lines without ARID1A and ARID1B mutations to identify negatively scoring genes, which can be putative drug targets. The second method we used to identify therapeutic targets was our own in-house genome-wide CRISPR knockout screen between isogenic cell line pair HEC1B, an ARID1B proficient cell line, and HEC1B BKO, a dual-deficient cell line that we generated in our lab. The resulting data was used to identify essential genes in the dual-deficient cell line HEC1B BKO. Using these two methods, we can narrow down the pathways we can potentially target, and we can validate these hits by treating a panel of endometrial cancer cell lines with drugs that target these putative essential pathways. These are the results of our DEP map analysis, where we are only interested in the negatively scoring essential genes that I highlighted in blue here. When a gene ontology enrichment analysis was run, we can see that the top essential genes in dual-deficient cells are involved in mitochondrial gene expression and mitochondrial translation. Similar volcano plots can be generated for our in-house CRISPR screen, where, like the previous analysis, we're only interested in the negatively scoring genes shown here. Specifically, we're interested in the differentially essential genes in HEC1B BKO, and when an interactome analysis was run on these 754 genes, we can again see that the essential genes are involved in mitochondrial translation and gene expression, suggesting that targeting the mitochondria may be a viable strategy in treating ARID1A and ARID1B dual-deficient DDEC. So we focused our efforts in targeting the main function of the mitochondria, which is energy production through oxidative phosphorylation. To validate our hits, we can treat a panel of endometrial cancer cell lines with drugs that target this pathway to see if the dual-deficient cells are more sensitive to these drugs. These are the endometrial cancer cell lines that we used in our study, split into two groups, ARID1A and B proficient and dual-deficient cell lines. We treated all of these cells with a drug called IACS, which is an electron transport chain inhibitor specifically targeting complex 1. However, because these different cell lines can have different mutational profiles, it can be difficult to elucidate if the drug response is actually caused by ARID1A or ARID1B expression. So we also re-expressed either ARID1A or ARID1B in a dual-deficient cell line, and we used the same isogenic cell line pair we used in our CRISPR screen to determine if changes in ARID1A and ARID1B expression can rescue or sensitize the cells to the drug. These are the results of our drug sensitivity assay for a panel of endometrial cancer cell lines, where you can see that the ARID1A and ARID1B proficient cells are less sensitive to the drug compared to the dual-deficient cells, as seen by the amount of blue here. And when ARID1A is re-expressed in the dual-deficient cells, you can see that ARID1A re-expression can partially rescue the cells from the effect of the drug. And a similar response is seen when ARID1B is re-expressed as well. Additionally, when ARID1B is knocked out of an ARID1B proficient cell line generating a dual-deficient cell line, we can see that dual deficiency can sensitize the cells to the effect of the drug, and these results overall suggest that ARID1A and ARID1B dual-deficient cells can be targeted with electron transport chain inhibition, and the response to the drug is in part mediated by ARID1A and ARID1B expression. In summary, this project will help identify new treatments for ARID1A and ARID1B dual-deficient dedifferentiated endometrial carcinoma, but we can take this one step further as ARID1A and ARID1B are not the only subunits that are commonly mutated in the Swiss-NIF complex. Around 20% of all cancers have Swiss-NIF complex defects, and I just highlighted some of the more commonly mutated subunits here. In particular, two cancers, small cell carcinoma of the ovary, hypercalcemic type, and non-small cell lung cancer, both often are dual deficient in SMARCA2 and SMARCA4. And we've shown in a previous publication that these cancers are also sensitive to mitochondrial inhibition, specifically the same drug that we used in this study. So there's potential that our findings can be applied to other cancers as well, and the study will provide preliminary data on targeting mitochondrial dependencies in other cancers with Swiss-NIF defects. With that, I'd like to end my talk here and answer any questions, but I would like to thank all the individuals listed here for all their help and support.